Before I begin, I want to highlight that I'll be making forward-looking statements, and I refer you to our recent filings for more information. Let me start by introducing you to the uniqueness of PASG Bio. Our mission is to provide life-transforming gene therapies for patients with rare monogenic CNS diseases that replace their suffering with boundless possibility, all while building lasting relationships with the communities that we serve. We put patients at the center of everything we do. We're focused on not simply identifying therapies for these patients, but on bringing them to commercial fruition. Our therapies must be transformative for our patients. CNS disorders are often devastating and catastrophic for patients and their families. Many of these conditions don't have available treatments, and they often lead to early death. A major part of what is so distinctive about PASG Bio is our collaboration with the University of Pennsylvania's Gene Therapy Program, or GTP, as we call it. I'll talk a little bit more about this in the following couple of slides, but suffice it to say that GTP is the research engine driving PASG Bio's pipeline, which right now, even at our early stage, is robust. At the heart of our relationship with GTP is PASG Bio's co-founder and chief scientific advisor, Dr. Jim Wilson, who is renowned in the fields of gene therapy and AAV technologies. Jim also leads UPenn's GTP. With GTP's cutting-edge science, we believe we have a differentiated approach that enables us to have a higher probability of technical and regulatory success. And because we can draw from GTP's prolific pool of cutting-edge science, we are also able to further de-risk our efforts by selecting approaches that have been shown through preclinical research to have the best chances of working. We have multiple potential value-creating clinical catalysts in the near term for our 3 lead programs. I will share more about each of these programs later on in our presentation. In further describing our distinctive value, I'll also mention the considerable time that we have dedicated to establishing our manufacturing capabilities. We have secured clinical scale manufacturing capacity through early commercialization for our gene therapy programs under an agreement with Catalent, and we expect our dedicated manufacturing suite to be functional by year end. In addition, after recently completing our IPO, PASG Bio has a strong balance sheet that will support our business into 2023, enabling us to drive our clinical programs forward to completion. Turning now to our leadership. As a result of our extraordinary pipeline and relationship with GTP, PASG Bio has been able to recruit a world-class leadership team with extensive experience in the successful development, manufacturing, and commercialization of gene therapies and rare and neurological disease products. Our deep bench of incredible leadership comes from both large pharma and small biotech companies with significant experience in running companies, clinical development programs, manufacturing, as well as negotiating and managing partnerships and business development transactions. I want to specifically mention our founders. Steve Squinto, who's our acting head of R&D and venture partner at OrbiMed, previously served as our interim CEO. Steve and I launched the company in early 2019, and we laid the foundation that has brought us to where we are today. Another co-founder is Tashi Yamada, who's the chairman of our board. Tashi is currently a venture partner at Frazier Life Sciences and formerly served as chief medical officer of Takeda and head of R&D at GSK. And of course, our third founder is Jim Wilson. Our CEO is Bruce Goldsmith, who came to us from Deerfield and also has significant experience in operations and as a CEO at other small biotech companies. Our accomplished team has been working with Penn to create a differentiated overall pipeline. As I mentioned earlier, PASG Bio has a unique and productive strategic partnership with Penn's preeminent GTP. This successful collaboration has resulted in a deep portfolio of gene therapy product candidates and also provides us access to future improvements in capsid engineering and next generation capsid libraries, vector engineering, transgene design and gene therapy modalities, as well as animal disease models and related studies for lead optimization of product candidates. Through GTP, we have the option to license 17 programs focused on rare monogenic CNS diseases. We have already licensed 6 of these programs. Consistent with Passage Bio's commitment to excellence, our impressive pipeline is the result of a rigorous and comprehensive development and evaluation process leading to selection of the best therapeutic candidates based on optimal route of administration, optimal capsid, transgene, and promoter selection, the ability to leverage mechanisms such as cross-correction effect, preclinical safety and efficacy data, and the availability of measurable predictive biomarkers. We also work with UPenn's Orphan Disease Center, or ODC, which helps to mitigate our risk in terms of clinical development relationships, as well as patient advocacy group interactions. The ODC is also performing a natural history study for GM1, which will be part of our comparative data set as we move forward from a registration perspective. Next, I would like to transition to talk about our deep portfolio of candidates. As you can see here, there are 3 programs in late IND enabling development. PGM01 for GM1 gangliosidosis focuses on correcting the beta-galactosidase gene defects of GLB1. We filed the IND in June this year. This IND is currently on clinical hold, and I'll share more about this later in the presentation. Notwithstanding this, our expectation is that we will be able to initiate a phase 1/2 clinical trial either in the 4th quarter this year or early in the 1st quarter in 2021. Our next program focuses on frontotemporal dementia, specifically on GRN, or the progranulin gene. We are planning to initiate a clinical study for this program in the first half of 2021. Our Crabbe program will follow a similar timeline. The therapies from all 3 programs will be administered via intracisterna magna, ICM, to provide the most direct access to the CNS. We also have 3 additional programs in MLD, ALS, and CMT2A that are all in earlier stages of development. Today, our presentation will focus on our 3 lead programs, but as you can see, our pipeline represents a balance of pediatric and adult rare monogenic CNS diseases. Turning our attention to our lead programs, I'll begin first by discussing GM1. With GM1, there's both infantile and juvenile forms. At Passage Bio, we are focusing on the infantile form, which has the highest incidence. GM1 is a disease caused by GLB1 gene mutations that cause a spectrum of disorders, including reduced muscle tone and progressive CNS dysfunction. Unfortunately, the infantile form progresses rapidly, with a life expectancy ranging only from 2 to 4 years of age. On one of the first slides in this presentation, we showed an image of a patient with GM1. You could see that this patient had a significant amount of supportive care, including ventilation and a feeding tube. This is why we're putting these patients at the center of our discovery and development program, because these are the patients that we believe we can help the most by intervening at the earliest stages possible. This is a theme that carries through to patients with FTD and Crabbe as well. The incidence rate for GM1 is approximately 0.5 to 1 in 100,000 live births, which translates into approximately 40 children born with this disease in the United States each year. We think based on external benchmarks and other analyses that this is probably an underestimate because there are no approved disease-modifying therapies and there is currently no newborn screening for this disease. For our clinical trial program, we have chosen a next-generation HU68 capsid. We selected this capsid because we have seen through preclinical studies its ability to transduce both central brain and peripheral tissues, which are both critical for GM1. The ability to choose the optimal capsid also demonstrates a key benefit of our relationship with GTP. Unlike many other companies, we can test different capsids in preclinical studies to choose the best ones suited for our purpose. As I mentioned earlier, we received a clinical hold letter from the FDA for our GM1 phase 1/2 clinical trial. We are in ongoing discussions with the agency regarding its feedback. We won't share specific details about those ongoing discussions, but we can confirm, as we've previously disclosed, that the clinical hold issues are limited to the biocompatibility of the ICM delivery device. We continue to be confident that our device will clear FDA's biocompatibility requirements and that we will receive a safe to proceed notification from the FDA to support initiation of our clinical trial program, either by the end of fourth quarter this year or early in the first quarter of 2021. We also expect to report initial 30-day safety and biomarker data late in the first half of 2021. Moving forward onto our investigational therapy for frontotemporal dementia, or FTD, caused by a deficit and deficiency in progranulin. Similar to GM1, where you see a decrease in enzyme activity of beta-galactosidase, here we're seeing a decrease in progranulin, which is a protein. FTD is one of the more common causes of early onset dementia, and about 5 to 10% of FTD, which is about 3,000 to 6,000 patients, is caused by a granulin gene mutation. And like GM1, this disease progresses rapidly, in this case to immobility and severe behavioral changes, including loss of speech and expression. After the onset of symptoms, survival is approximately 8 years. This is a rare, underserved disease, and there's no approved disease-modifying therapies today. As with GM1, for FTD, we selected the capsid that we believe is optimal based on preclinical data, which in this case is AAV1. The observation that really drove our selection of AAV1 was that we saw the progranulin expression of up to 50 times. That's 50-fold higher than what is found in normal human CSF, and actually 3 to 5 times greater than what we see with the HU68 capsid. We are planning to initiate our FTD clinical trial program in the first half of 2021. The final program I'll talk about today is Crabbe, which is a lysosomal storage disease. We are using a very similar approach and overall strategy to GM1. Unfortunately, this is an especially devastating disease, particularly for the infantile form of Crabbe. The disease progression is highly predictable and includes loss of acquired milestones, staring episodes, apnea, peripheral neuropathy, severe weakness, unresponsiveness to stimuli, seizures, blindness, and deafness. It also progresses rapidly with mortality by about 2 years. The estimated worldwide incidence is a little bit higher than GM1. Our work has found that the estimated incidence is likely around 2.6 per 100,000 live births, which translates into almost 100 children per year in the US. With this disease, we are focusing on the GALC enzyme using a next-generation HU68 capsid. In preclinical models similar to GM1, we observed meaningful transduction of both CNS and the periphery. Our plan is to initiate a clinical trial for Crabbe in the first half of 2021. From a financial perspective, we have a strong cash balance of $353 million as of June 30th, which gives us a runway to potentially deliver multiple meaningful catalysts over multiple programs. Passage Bio is not only committed to advancing its own pipeline with a goal to be the world leader for rare CNS gene therapies, but also to be a highly valued and engaged partner with patient advocacy groups and the larger gene therapy community. Our commitment is to share the groundbreaking research fueling Passage Bio's pipeline with the larger community through publications and presentations. Our vision is that the promise of gene therapy is finally fulfilled for patients with rare CNS genetic diseases.